So, as Ko-Wei said, I'm going to talk today about a couple of projects that our group has been involved in, both of which make use of knowledge graphs. One is the NCATS Biomedical Data Translator, and the other is the Common Fund Data Ecosystem Knowledge Center. And I think one of the main reasons that there's interest in this group, I gather, is how they've approached this question of knowledge graphs in biomedical research. So I'm going to give some thoughts on how they've each addressed and come at this question, and also give my thoughts on, I think, what it implies for the field and the challenges that we're currently facing in making knowledge graphs work the way I think we all want them to work in this area. So before I do that, though, I just, almost by way of disclaimer, I want to give just a little bit more on my background and the projects I typically work on, because I do not actually work in knowledge graphs myself. Most of the work that our group does with Noel Burt's group at the Broad are building genetics and genomics knowledge portals. And you may be familiar with the type 2 diabetes knowledge portal or the common metabolic diseases knowledge portal, both of which are funded by NIDDK and part of the Accelerating Medicines Partnership. And beyond this work, I do research. Our lab at Boston Children's is a statistical genetics and computational biology lab. So we are, for example, very interested in statistical calculations of the extent to which genes can be linked to various different diseases. And so, this is the type of data that we typically are producing and working with. This is like a plot here that's showing for one gene statistical estimates of how linked it is to thousands of different traits. And so this is the type of work that we typically think about, which is very, very different from the discrete, kind of assertion-based world of knowledge graphs. And so that's kind of like almost by way of a disclaimer that I'm going to give you my thoughts today on knowledge graphs and these two projects in particular, but it's coming from the perspective of a statistical geneticist. And whether you think that's a strength or a weakness, I guess, will depend. I'm not going to talk really about the challenges in constructing these, but I think I'd like to think I have had a lot of experience with using them, and essentially the opportunities and current challenges we face in making them useful for things as basic as what could be some novel drug targets for a disease of interest. And that's really what I'm going to talk about today. Okay, so here's my basic view at like a 30,000-foot view of like where these two projects stack in terms of, if you think about the knowledge graph landscape. So I kind of have here the Google model and the Netflix model as two different ways that these companies have kind of attacked, have two kind of different business models. So Google began with like a very broad and like infrastructure level type of mission, which is let's organize the world's information, and then let's figure out later how to monetize that, or what applications can follow from that. It turned out there's quite a few, right? The other end of the spectrum, you have Netflix, which started as like a DVD mail delivery service and now has since kind of found their niche and gradually expanded it to now they're like a big media company, right? And the way I place kind of Translator and the CFD Knowledge Center are coming at knowledge graphs from like these two different angles. Translator is a project that is much more focused on infrastructure and building things that are necessary for knowledge graphs to work in terms of biomedical queries. Whereas, as I'll talk about for our work on the CFD Knowledge Center, we've taken the much more very specific approach, hoping to generalize it later. And I think kind of the punchline at the end will be, I think that there's value to both of these approaches, and maybe by kind of combining them, we might arrive at something that I think is really combines the benefits of both of these approaches and can address some of the important questions that are outstanding. Okay, so I'm going to start with the Translator project. I'm not sure how many of you are familiar with this project, so I'll give you an introduction into it and then kind of demo the resource that currently exists for this, and then talk through some of, what I think some of the interesting challenges that we're currently facing. And these are slides that I borrowed with permission from Tyler Beck, who's our program officer at NCATS, who works very, very closely and has been a key leader in this consortium, particularly over the last five years or so. So Translator's actually been around for a while. It says here 2017. I feel like I remember working on it starting in 2016, but it's in its third phase now, actually, that just started, I think this year. So the initial phase was feasibility, where there were 11 different teams who were evaluating whether or not something like Translator could be built. The second phase was building out, like really is where the knowledge graph and the infrastructure was built. And now we're beginning the third phase. This slide's out of date, which is really getting at how to, it's called the performance phase. It's about making the work that's done scale. So what is this work? So there actually was originally a very specific question or area of work that was proposed by Chris Austin, who was the director of NCATS about 10 years ago. And it was this observation that clinicians and basic scientists think about diseases in opposite ways and opposite terminology, right? So basic scientists are working on molecular and cellular processes and data collection at that scale. Clinicians are describing diseases in terms of symptoms, but if we want to develop better treatments, we need to have a way to link and translate, right, between these two different languages. And so it was this play on words, that translational science, but also translating different terminologies. And so this was like, when we applied for this grant, it was really we thought this was a very interesting question to address. But translators actually evolved very much so more towards infrastructure building. So this was the slide that really, the most recent one that Translator built that describes the goal of the project. And you can see there's a lot of things here about infrastructure for data-driven translational research. There's a huge focus on taking many different data sets scattered around in different silos and allowing them to be cross-queried and reasoned over them. Open data, open team science is a really big focal point as well. So Translator has really, if I had to frame this in terms of thesis, antithesis, synthesis. So thesis, Translator really takes a hard claim that knowledge graphs are really essential infrastructure if we're trying to realize this vision of open data in translational research. So it's, like I said, very focused on the appropriate way to link data together and make it queryable and make it open and available to everyone in the world. And that's where most of its progress has been achieved over the last 10 years in establishing those sort of things. So what does it look like? So this is like a schematic of how Translator works. So basically all of the data from various, actually, maybe I should start with like the data. So like these are examples of like the type of data sets that have been integrated into Translator. They include things like, that are text mining from like PubMed, but also these databases like Drug Bank, ontologies like gene ontology, basically all these different public data sets that are not necessarily interoperable. All of those are, onboarded into Translator, and linking amongst them and querying over them is really the goal. And this is the architecture that has emerged. So essentially, all of these different data types get organized within a knowledge graph. It's not actually like a single knowledge graph. It's like federated. And the way it's federated is that we have these different knowledge providers. Each knowledge provider is like a team who maintains a REST service that they're responsible for curating a specific type of data, and they make it available as part of this like virtual knowledge graph via these web services. And then these reasoning agents then can query across all of these different knowledge providers, and based on what they get back, they can respond to different queries and come up with, relationships between two different entities. And I'll get into a minute of what that looks like. But these are doing like the brunt of the AI work, quote unquote, where they're reasoning over these graphs that are kind of kept federated at this knowledge provider level. And then you have this orchestrator at the top that like kind of interacts with the user, and the user will ask a question and it will like spin it down to these reasoning agents, which will kind of query the knowledge providers, et cetera, et cetera. And so that's kind of, what the architecture that's emerged. And in order to make that happen, Translator has done a ton of foundational work on allowing this type of architecture to be possible. So here's just kind of some major technical achievements that have come out. So the BioLink model is basically a schema and an ontology that kind of unifies all of these different data types. And there's been a ton of work as part of the translator community in essentially allowing you to harmonize across not only these data sources, but existing ontologies that exist as well. Entity resolution, so matching up names across different resources is a big thing they've done. This TRAPI, this is a standard for how you can have something like a user query at this level be dispatched to these different reasoners that can then request things of the knowledge graph. So it's this very generic way to allow you to kind of query across at a very abstract level all these different knowledge providers and knowledge graphs. And then all of this is done like in an open way via this what's called a smart API registry, which is where all the tools are registering. So there's been a ton of infrastructure, in other words, that have been built for allowing these type of, many different data sources to be put alongside one another and harmonized and to be queried. And that's really, I think, one of the major advances of Translator. So as I mentioned, I do not work in any of this stuff. Our group is part of one of these knowledge providers. And in particular, our role is to onboard this type of genetic data that I showed you within Translator as the genetics knowledge provider, right? So we take, so it's like one example of the data we have. This is like showing a plot for SLC2A2. It's a gene many of you have probably heard of. And we have all this information that we've distilled either common variants from GWAS into relationships between that gene and a disease, or we have rare variant data as well. So we have all these statistics. I'm not going to go into the details too much there, but we have to figure out a way to get that into the knowledge graph so it can be used by Translator. So we got to boil down these complicated statistics into edges that, like in the BioLink model, will tell you here's a probability of disease relevance between this gene and a variety of traits. And that's really what our goal is as part of Translator. And for those who've worked with knowledge graphs before, you won't be surprised. It's quite complicated in practice, even though conceptually it's simple. In practice, there's a ton of work that needs to be done. And this is actually what a single edge of that looks like in the BioLink model. So you can kind of squint here, but you have an object, which is your gene. The condition associated with gene is what BioLinks allows us to provide a link to the subject. In this case, this is a disease. And I forget what Mondo this is, what disease that is. But that's basically what it looks like that we're contributing to Translator. And then our data kind of gets folded in alongside the other KPs. And then a user interface can now, users can query it via the Translator user interface. So I'm going to now demo that. So maybe now's a good time. Are there any questions about any of that before I kind of show you what it looks like to a user?
For all the participants, you can use chat function or just unmute yourself if you have a question right now.
Feel free, please, just to send chats along the way, and we can pull those up at any point, just as they arrive at you.
Mm-hmm. Yeah.
Okay.
Mm-hmm.
So here's like an example of a query. Okay, so this is what drugs can possibly treat chronic pain, and this is kind of like what the query looks like, where you're essentially constraining. You're looking for edges, treats, which is your predicate, and then you're constraining the object, which is chronic pain, and you're looking for things that match chemical entity. And then this is showing kind of like what this would look like in TRAPI. But fortunately, you do not need to use TRAPI or anything like that. You can go to the Translator user interface, which is at ui.translator.io, where there's no vowels at the end for some reason. And let me just show you what that looks like. So right now, the way that Translator does not let you just like explore like the full knowledge graph of all of its nodes and edges. We're working on queries that allow you to just say, here's two entities, can you find paths between them? But that's quite computationally difficult. So right now you're restricted to only one of 4 different queries, okay? And what this one is, what drugs may, I guess there's 5. What drugs may treat conditions related to is what we call the treats query, which is one of like the more, kind of more important ones, as you can imagine. So if we show like chronic pain, and I search, I guess I'll look at the disease. So what's happening now is that query got dispatched to that ARS, which is the head of like the whole reasoning system. It dispatches its results to all the ARAs. They either have things cached or they are sending things to the knowledge providers. And then the results all come back. And then there's all this combining and ranking and sorting into like this integrated result list here. And it comes back asynchronously. So down here, the annotate button's hiding it, but I think it says some number that are added. And as you see, as new stuff appears, you'll have an option to like sync new results and they'll kind of update automatically. And then there's ways to filter results that are coming back. I'll show you that in a minute. But for each result, so this is showing us, we constrained drugs may treat conditions related to chronic pain syndrome. So each of this is going to be a drug. And you can expand these to see the reason that this inference was made. Okay, so right here, this is giving you evidence for this assertion that ketamine can treat chronic pain syndrome. And this is telling you the reasoning paths and like the publications and sources. So if you kind of unpack this, you can see that there are what are called indirect edges that are multiple of these paths that are supporting it. And if you click on each of these, you'll get information into the provenance. So this particular assertion came from one of the ARAs that's called unsecret agent. And then you can drill down even further and see what evidence sources unsecret agent used for this. So in this case, it was a lot of different text mining from PubMed, these more publications, I don't know exactly how many, that are linking you to the specific assertion. And then this is telling you where that assertion came from. So text mining targeted association API is one of the knowledge providers. So this is basically an inference that's being made by one of the ARAs, unsecret agent, using information from the text mining knowledge provider. Okay, and you can see that there's other ones. Down here is maybe a slightly more complicated relationship where, again, in this case, the evidence is coming from the toxicogenomics database, but there's also multiple different supporting paths for that, right? So it's coming from this indirect query where you can see ketamine treats fibromyalgia, which is a subclass of chronic pain syndrome, as well as kind of this direct link. So all of this is kind of like done for you as the user. It's like a complicated set of paths. They're all scored, they're all combined, and all of these are things that you can browse. And on the left is where you can start to, filter and refine the results that you're being shown depending on what you're interested in, right? So if you don't want to see like unsecret agent, you can hide those, or you can only focus on another reasoning agent. This Aragorn is another one. You can constrain the types of paths, right? So this will require, paths to have a gene in them. All right. So if I don't want just the direct things from the literature, this will require there to be a path that has gene as an intermediate, right? So now this is telling you that gabapentin was picked because it targets a gene, or it affects a gene that was targeted for fibromyalgia. And again, you can see the same type of provenance for all of them. And so, like I said, translator, like we are, the consortium is very well aware of like the performance challenges and the UI challenges. And that's really what a lot of the effort has been spent on. And in the current phase, making this so it's much more efficient and fast. So, we're not only 45% loaded after however long I've been showing this to you. See some stuff in the chat. Can you address data quality and tests for poor quality data or data mislabeling? Yeah, so that's supposed to be the realm of the knowledge providers, right? So in principle, the knowledge providers are the deep data sources for translator, so we're an expert in genetics, so we are responsible for assuring that we have high quality genetic data, and that we handle that the data is not mislabeled and whatnot. Now, having said that, as we'll show in a minute, that doesn't handle integration across different knowledge providers, which is a major challenge I'm going to discuss momentarily. Can you explain more about why using a KG is important? Why is KG so special? So it's an excellent question, one that I spend a lot of time thinking about, and I will discuss that in partial length very shortly. I think that the rationale for why translator thinks and why knowledge graphs have taken off, not just in this consortium, but in others, is that it is one of the few ways you have, it's one of the few abstractions you have of taking data sets that on the surface have very little to do with each other and representing them within a common language. So if your goal is to like unify human genetics with chemical compounds, it's like you need some type of universal language that can map between them. And knowledge graphs are a very flexible way that has enabled you to do this. And when you combine them with ontologies, that allows you to do things like reasoning. And then the knowledge graph also gives you a chain of evidence. So a lot of that's one of the reasons they've been around for quite some time. And I think one of the arguments for using them in these challenges of like harmonizing disparate data. Having said that, I think there are some definite considerations that need to be made for the specific application to biomedical data, and I will be discussing that at great length, well, not great length, but at length momentarily. What if they make an error? That is also an excellent question, and you'll see in a minute. I think the answer is they're unfortunately, knowledge graphs are a little bit brittle to that, and I will give some thoughts on how those errors actually are happening and making it hard for us to, one of the current challenges facing translators. So I'll return to that in a minute. How we can make it implemented in type 2 diabetes research. Is that question how you would use the current tool for type 2 diabetes research, or like actually how this would be, okay. So for now, and you can see, so navigate away from the current page. So type 2 diabetes is a query you can run, okay? And if you ask what drugs may treat conditions related to type 2 diabetes, you will get evidence for this, right? And you will be able to see what translators have been able to pull in. And presumably the goal is to make this type of query relevant to someone doing type 2 diabetes research, right? This is not for clinical providers, right? To actually find drugs to administer to their patient. It's to try to come up with novel hypotheses about how new drugs or mechanisms that you might target for type 2 diabetes. Now, if you type in your favorite disease, you're going to see, this will be a segue. What you will see is that the results are not necessarily what you'd probably be hoping to find out of a translational research tool. Not sure when this is going to come back. I'll give it I'll give it until I'm finished talking. But essentially, okay, so here we go. Like what you'll see is that most of these top results that are coming back are all like very well-known drugs. And you can get around this to some extent by like requiring, like I guess these haven't come back. Like there is a way to basically say I only want things that go through a gene, right? So what came back first were the results that are all just single hop queries that are just direct lookups of Drug Central, right? Which is probably not what you want. You can kind of constrain this a little bit more to say like, I want to only have, indirect queries, and then you'll find things like up here that at least, but, one of the things though, the more you look through this, I think these questions are already getting at the point I wanted to make. It's, before you even saw these results, which is that unfortunately, most of the top ranked results you're going to find are going to be dominantly driven by text mining. And in fact, things that are just broad associations between your trait and a compound, as opposed to probably the biological model that you have in mind, right? So for me, for example, what I would want to find for drugs that could treat diabetes are those that have genetic support, right? That there's like a genetic variant in that gene that is associated with type 2 diabetes. And there's some other stuff translator can layer on about the mechanism of action that it together can tell me, right? But so in other words, I want this information kind of married alongside kind of these more straightforward direct one-hop things. But it's like very hard to like get those results right now. Right now, you may have a different like thing that you want, but in principle, you'd ideally want to be able to like kind of explore this in a more flexible way that's kind of getting at your hypothesis, right? And Right now, like some of that is performance issues, and like, but a lot of it I think are fundamental challenges we have to address in the knowledge graph field. And so like, so I think, I'm not going to, I want to go to the other resource and not spend too much time here, but it's all part of the same theme. So what I'll say instead is the specific problem that we have faced in this project is that our genetic data is like never used in any of the queries. So it's just not possible for me to like do what I want to do, which is find me genes that are associated with diabetes that have additional support from all these other data sources for translator that might be promising drug targets. And there's lots of reasons for this, which I can go into time permitting, but I'll just list a couple here. So one is that a lot of the data sources that you want to use in biomedical research are statistical inferences from really complex, noisy data, which can be encoded as graph structures, as I just showed, but discard a lot of information, right? There's a big difference between, like, this is actually, we'll get into a minute how these are calculated, but it's this very complicated model of like the underlying way in which like genes manifest associations that has to be completely stripped out as soon as you force it into the BioLink model, right? And then because the generality of knowledge graphs is a double-edged sword, because then the reasoners, in order for them to reason over all these different sources, have to almost by definition not be specialized to any one evidence type. So they don't know that these genetic data sets are special to me as a user, or might have like a model underneath them. And then, so what's happened with this project is that over time we identify these issues, but the way we address them is, okay, we need a better scoring system, or now we need a way to like filter better. And so it's almost like playing whack-a-mole, whereby you are trying to overcome this issue by building things on top of it, and it eventually gets quite complex. So what that I think this is like kind of an image of where my take on this is, where we stand with Translator, is that our goal is to have this knowledge graph that represents all this beautiful knowledge that is across all these different sources and is this great abstraction, allows you to do all this incredible AI over it. But where we currently are is that just in practice, like the knowledge sources that are easiest to onboard and the trade-offs you have to make with reasoning kind of gets you basically a bunch of text queries that are prioritized, and it's non-trivial to get the other data sources in there. So I think that's the challenge that I'm really interested in working on in this project, which is like, how do we start to surface some of those other data sources, right? Because I think if you just have a query results that are basically lookups from text, you now have like a completely other elephant in the room you have to compete with, right? Because LMMs can also query PubMed and they can query text, right? And there's billions of dollars being invested in those. And so if we're trying to like address this problem for biomedical research, we want to put where we have special advantages. And to me, it's these questions that just came up, right? Which is, how do we ensure that like curated and high quality, and the results that you get back are kind of robust to failures, and address questions that scientists want to think about. And so that I think is, so if go back to my narrative, infrastructure out, where we've ended up with, I think, is infrastructure that I would recommend anyone using who's going to do a knowledge graph, because there's pretty much everyone who knows a lot about semantic web and all these ontologies has contributed Translator. It's a very general standard. If you represent your data in it, you are immediately hooked into all these great resources, right? So the infrastructure is like top notch. But application, now we end up with, it's so abstract that we now need to start thinking about how can we distinguish the applications from things you can do with an out-of-the-box LMM. And I'm going to put a pin in that because I'm going to come back to that after we talk through Common Fund. So any questions on that beyond what we had in the chat already before I move on to this? How am I doing on time? Okay, I'm okay.
Yeah, I don't see other questions.
All right, great. Okay. So now going to come at this same type of question from a completely different perspective, and that's through the CFDE Knowledge Center. So many of you probably don't know what this is. So let me unpack it in several steps. So the first is, what is the Common Fund? That is an NIH funding mechanism that supports basically very high impact, cross-cutting, very important foundational projects. Okay. The Common Fund, like, you may not have heard of this, but you've definitely heard of a lot of the things it funds, like GTEx, HubMap, Kids First. These are all like huge projects that are get in major journals and produce incredibly valuable resources. So the Common Fund is an incredibly valuable source of data. And the Common Fund data ecosystem was formed to try to leverage that and to try to like make these data sets interoperable and reusable, right? So very similar to the translator mission, but in this case, ironically, if translators started with a biological query, Common Fund started as an infrastructure. What's the interoperability that we can, how can we make these data sets interoperable? So each of these bubbles are one of those CFDE programs or DCCs, depending, I don't know what I'll call them, both of them are synonymous. And then there's 5 centers as part of the CFDE. Okay, so the ecosystem is like this center thing that is supposed to integrate across these different programs. And we are the knowledge center. So we're tasked with kind of extracting knowledge from each of these programs and making that interoperable. There's also a data center where you can download data, and then there's some administrative stuff. So the challenge here is basically similar to the translator one, right? You've got these like data sets that on the surface have nothing to do with each other, right? So like if you're familiar with the LINKS project, like you perturb cells with either knocking down genes or knocking out genes or hitting them with chemicals, and you get like readouts of gene activity, right? That's the LINKS project. It has nothing to do with A2CPS, which is a study of chronic pain, or like HubMap, which is like spatial single cell omics data, right? So nothing to do with each other, but we're tasked with trying to integrating them together. And now this is back to like that earlier question. Knowledge graphs are one of the logical ways that you can do this, right? Because for any one of these programs, you can manually encode, or you can write a script to encode, if you're an expert in it, your information as nodes and edges in a graph. And then those can all be unified. And there actually is a program, a project within the CFDE called the Data Distillery that has done this. And this actually is completely unaffiliated with, has nothing to do with translator, and actually is like a non-interoperable technology. I think you might be able to make it interoperable, but it definitely does not use like Trappy or BioLink or anything like that. Uses this thing called the Unified Biomedical Knowledge Graph standard, which came out of UMLS. And this is much more of like a bottom-up approach if translators top down. There was like a very small group of people, Deanne Taylor and Jonathan Silverstein, who really took the lead on this project and worked with all of these representatives from these sites to encode their data in it.
Then there's a question.
Will the LMM be incorporated in the translator in the future? Yes. So that is something we're actively talking about, and what I think is the right answer long term. So I will talk about that briefly at the end. But you'd expect more LMM use. There's like a lot of people are in transit are using LMMs, but right now it has not yet been used, which to me would be quite obvious as a wrapper outside of the UI. I know people are thinking about that, but it's not like planned for any time in the near future, but definitely something that will almost certainly happen. Okay, so all right, so this is Data Distillery. Now, when we applied for the Knowledge Center, we actually took the complete opposite approach. So we took the Netflix approach, which is let's build very specific, bespoke, or dedicated integrative workflows or queries that are answer like one question, and let's build a bunch of them. And then like over time, let's try to figure out how to generalize these more. So we called these like secondary knowledge sources, which is like a single Common Fund program. You can access data that wasn't published, right? Whereas these more complicated cross resource queries we called tertiary knowledge. This is based off of like an analogy with like historical sources, like secondary or tertiary. And so you can think of this as like the CFDE, our approach at least, is like the antithesis to translator, right? Like don't build out infrastructure for infrastructure's sake. Start with like very specific questions and then solve those really well, and then grow your resource over time to take on more and more and more. Okay, thanks. All right, good. All right. Okay, so let me show you an example of what that looks like. So the URL for CFDE, our knowledge center, is cfdeknowledge.org. And that is here. If you come here, you'll come to the home page, and there's all sorts of information, which I'm not going to dwell on. I'm going to show you instead like the gene page, which kind of gives you examples of some of these secondary knowledge sources. So each one of these cards is a analysis that we had to meet with the DCC to understand and implement a method and design a visualization that only answers this one question. So for GTEx, for example, this is a measure of how specific expression of the gene is to all the GTEx tissues. And you can think of this as like, it's very limited in what you can do, but we are targeting it for people who aren't familiar with what these common fund programs offer. This gives you a way that you have your favorite gene. Many people do. You can type it in and you can see what the common fund resource says about it, which might then get you more interested in like navigating to the full GTEx portal or downloading more data. And it's kind of like the entryway into it. Are you building query to Spark in form of the SCS? So Spark is one of the ones that we have not yet onboarded, but we actually worked with Marianne on another proposal, and it's something that we're actively thinking about. But Spark has kind of been difficult for many of the people in CFDE to integrate based on just how different it is from the other data sources. But you're right. Like that type of schema would be quite useful. And so like another card, this is, I guess, doesn't have data for PPR gamma, but if you search for like a gene like IRX3, this is an example of 40 nucleome is like the 3D structure of the genome across various time points. So it allows you to link faraway regions of the genome to each other. So this is telling you what variants are linked to IRX3 that are associated with other diseases. So kind of get the idea, right? It's like this is a very, very hyper-specific analysis that we're doing, and we have them for every, common fund program on here. And they're not interoperable, they're not linked together, but they are at least like pulling out value and information from these resources that can hopefully, at least, even if they're not yet addressing interoperability, they can address reuse, right? And so these slides just kind of go through examples like, there's, I think, 10 or so of these on the gene page. But of course, reuse is just one part. We want to make them interoperable, right? And as I mentioned, that's a big challenge. And one way is through knowledge graphs. But another approach that we are taking is, you can think of this as a complementary language for unifying across different data sets. And that is the language of gene sets. So I'm not sure if anyone here works with gene sets, but essentially it's very simple. It's just a set of genes that you can generate. And ostensibly, the gene should have something in common and capture some type of like biological process. So an example is, here's a gene set that is in the Data Resource Center that is genes that were upregulated after 8 weeks of exercise in the blood of rats. And that's from the MotorPack Consortium, which is a common fund program that studies the effects of exercise at the molecular level. So they have like a bunch of gene expression readouts amongst all these rats at like different time points. And you can take one specific readout, which is what genes are upregulated after this amount of exercise in this tissue, and you get 689 genes that are the most upregulated. And that's like, we have no idea what that's capturing, right? But presumably it's capturing something that happens to the blood after exercise, right? And you can do this over and over and over again, right? So it's very scalable. It's much more scalable than generating assertions, of course, much more limited because you're restricted to just bags of genes, right? But, this has been done. Our colleague Avi Mayan, who is the leader of our sister resource, the Data Resource Center, has curated about 37,000 of these. And then once you have these sets, you can do things like intersect them to find like significant gene sets that match to each other. And so it's very scalable. Now, the issue is that you have no idea what they mean, right? And because there are so many of them, it becomes just this complicated thing. Like, what does that gene set mean anything? How does it relate to, anything that I care about biologically? Which ones are relevant to traits of interest? And the way this is commonly done to interpret these gene sets is that you compile this library of like very well-known gene sets. So like if you're like wiki pathways, right? It's like a set of gene sets that are tagging well-curated biological pathways. If your gene set overlaps that significantly, that tells you that you're capturing like a pathways, and that's telling you what your gene set is doing, right? So this concept is called enrichment, and Avi has been one of the leaders in that and has been working on this for quite some time. What we have done for the Knowledge Center is to do a very specific type of enrichment, which is checking overlap between CFDE gene sets and human traits, okay? And so you can take a GWAS for type 2 diabetes, and you can get the genes that emerge from that. That's how you do that. Is not trivial, right? If you just take the GWAS catalog nearest genes, you're not capturing most of the information. But in principle, that's really what we're doing with our Knowledge Center. And to make this much more statistically rigorous and principled, we have this statistical method called Pigeon that I'm not going to talk about, but you can see a webinar of it at our Association to Function portal. And there's also a web portal here if you want to record that, that you can access the results that have nothing to do with CFDE, but are just applying this method to a bunch of different traits. I'm showing this figure not because I want you to understand how it works, but just how complicated it actually is to design a model that does something as simple as link a gene set to a human trait. Okay, like you have to model the gene set down here. You have to model genes that are in the gene set. You have to model SNP associations, and you have to like encode how SNP associations change as gene values change and how gene set value. So there's a ton of work. It took like 2 years to develop this model. And there's a ton of like tuning and statistics that go into it, right? But once you do this, it's quite valuable because what it outputs is very straightforward assertions amongst gene sets, phenotypes, and then the genes that are in them that can be treated as assertions, right? But now the assertions are very statistically calibrated, that you have values that matter that can be like interpreted probabilistically and are not just, this question earlier, like, how do you know if there's a mistake? Well, this is like mathematically provable of what this is producing, right? And you have very, very strict and, controls over them. I'm glossing over a lot of details, but hopefully the point that there's a really, really deep statistical approach taken to generate these higher level assertions is coming clear. Is this a causal graph? No. So the graph is not, well, so this is not a causal graph. This is what's called a Bayesian network, where this represents conditional independent assumptions, which are not necessarily causal. You could make an equivalent graph with arrows in the other direction. So there is causality coming in, but it's via human genetics, and I'll kind of omit that here today. Okay, but I want to show you like kind of what this looks like. So you can access this via this gene set analysis card on this homepage here. And this is going to give you examples of all of these different types of relationships. So each of these rows you can think of as one of those assertions, which is linking a gene set to a phenotype. And then also there are genes involved. And rather than kind of demo it live, I'll kind of go through one example for MotorPack and kind of walk you through the type of information that's encoded here. So I'm expanding here an assertion between the gene set, and I kind of described what these are, right? Genes that are, it's blood RNA male 2 weeks up, that's from MotorPack. It's associated with reticulocyte count or linked to reticulocyte count. And then these are the genes that are in the gene set that are also associated with reticulocyte count. So I guess the edge would be like you have phenotype linked to gene set, and then you have a bunch of genes hanging off that edge, right? And so walking through it, so what this is telling you is this is some measure of genes upregulated after exercise. So we know that the assertion has something to do with exercise, right? It's linked to the count of immature blood cells, and that's from the human data, right? There's some effect sizes, which I won't go into, but essentially can be thought of as actually are log odds of probabilities. And then it gives you the genes that are in the set that are also associated with the trait. So not every gene in this set is actually relevant to reticulocyte count, right? We just know that broadly there is an enrichment, but you can pull out the specific genes that are. So what this basically gives you is an inference, right? There's actually multiple. So first off, it tells us that that gene set, whatever it was, is interesting. Second, it tells you that they have a connection in humans to reticulocyte count, right? And it tells you the specific mediators. And to the extent we can say something about causality, we know that the genes have a causal role in reticulocyte count. We don't know that they have a causal role in like exercise, right? Or because the differential expression results are, you can't infer causality from, but the human genetics does give you causal link to the trait. So you now have like mediators that are causally affecting reticulocyte count that are upregulated after exercise. So there's a ton of information baked into this if you think about it, right? And obviously those who know anything about exercise, I don't, but basic searching, like obviously this makes a lot of sense. It's very well known that red blood cell counts affected by exercise. And the known mechanism is through stimulated erythropoiesis, which some of these genes are, but others actually are more like foundational for like red blood cell like stability and iron transport into it. So there might be some additional mechanisms at play for that relationship that you can glean from the genes. But like, as I said, like it's quite complicated, right? Like, and what we found when we put this up is that a lot of this is like too much information for a lot of people. So we've tried to even simplify even more and like make even more specific user interfaces that make even more specific questions tailored to people. And in this case, this is like a specific application of this method around mouse genetics, okay? So essentially, the way to think about this is one of the CFDE programs is the, see if I can find this, the Knockout Mouse Project. So what this did was generate a lot of gene sets that are the genes that when deleted in mice cause a specific phenotype. And so every link between that and a human phenotype is essentially, you can think of as a genetic match, right? We're finding a mouse phenotype and a human phenotype that share common genes, right? And this is very different from the way mouse and human phenotypes are typically matched, which are done based on semantic similarity. So this is the Monarch initiative, which is heavily in Translator. Now, Translator would do it. You're often matching phenotypes based on how similar they look. But one of the issues that mouse geneticists, I understand, get from grant review panels, but also things that could, be one of the reasons a lot of animal models fail to translate to humans is that phenotypic similarity does not always imply genetic similarity. So the hope is that maybe by finding matches between mouse and human phenotypes that share genetic links, you might be able to have more confidence that, they'll translate to humans. And so that's essentially what this like other module does. This is essentially just the exact same data I was just showing, okay? But now it's just hardwired, so you just type in like a specific mouse phenotype, okay? And what it's going to do is pre-filter your results to now kind of just visually show them in a more intuitive way of the human traits that are genetically linked to that mouse trait. So rare deafness is linked, I typed in head bobbing. So a lot of deafness diseases. But I think if you look further down, you'll see something, I think, related to like a bone disease or something. So some things that like are surprising, some things that are confirmatory, but, again, something that we're trying to think about targeting a much more narrow user group in terms of applications of like these types of methods. Okay, so now, so basically this is, I mentioned, the kind of the opposite approach of Translator, right? We're now very, very specific. One question you can answer. We designed our own method. We thought a lot about the UI to basically answer one question. So the hope is that you can reach more people, right, who don't naturally think in terms of graphs and base queries. But on the other hand, it's like not at all obvious how you would extend this to other traits, right? It's like very much more limited in scope, right? It's not achieving the full vision of CFDE, let alone Translator, of making data sets like fully interoperable. Okay, so kind of conclude, and actually, let me just finish. I have like 5 slides, then I'll stop for last questions. But so that to me is like, so the synthesis, right? Which is that I think there's a role for both of these approaches, which is statistical inference is a very valuable way to extract information that's high quality from the data sets that we have, right? But then you ultimately want to put those into a knowledge graph that you can essentially address the broader question of integrating and making things interoperable. And now you kind of have hopefully the best of both worlds. And I think that's particularly germane with the, I wouldn't say threat, but the elephant of LMMs, right? Because if all your knowledge graph is doing is onboarding stuff from public sources, you might say you're doing a better job than LMM, right? But like companies are investing tons and tons of money in this. That's going to happen. They're going to strip everything that's possible from public sources. The one thing that they can't access, and it's not even obvious to me if LMMs can handle, can be extended to this based on how they work, is complex data like gene expression signatures, right? Or GWAS associations. Those just fundamentally like cannot be modeled by LMMs, right? And so there's a huge opportunity to get those data sets extracted with high quality and onboarded into these AI systems, which could be knowledge graphs, but even if it's LMMs, you're still doing a huge service by only we as biomedical researchers can like actually get that data to be high quality. So I'm showing here, for example, that last method, Pigeon. This is like just kind of a very, very small subgraph of the relationships amongst gene sets, which are squares, and genes, which are these big circles, and traits, which are these small. So this is like a graph, quote unquote, but it's completely uniform and harmonized. And it's like the quality is very high from the statistics that we've developed for it that only we can do, right? And my guess is there's a lot of other examples like this that if you're an expert in a type of data, you can come up with ways that we can build these very high quality and quantitative edges. And then you layer on top of that graph the additional like traditional ontologies and schemas that Translator, for example, is doing. And then like the future is, okay, is that going into just reasoning agents, or are we doing some type of RAG-based approach? Or so RAG is retrieval augmented generation for those, where basically the LMM like queries either text, you give it text, or it queries a knowledge graph. And then it can like, on top of it, do its thing. So I do think that the future is like using KGs as part of LMMs. I always say LMM instead of LM, as part of LLMs, right? That's not my area of research, but I think what I am very passionate about is getting those edges that are locked away right now because they're in all these data sources, like very high quality and statistical. So that's kind of the synthesis, right? Standards infrastructure, highly recommend Translator. I do think more investment in bespoke methods for getting data prepared for that. And then going forward, yes, I think a combined approach is the best. And before I answer questions, just want to make sure I acknowledge the people who worked on the CFDE. I don't have a slide for Translator, unfortunately. I want to make sure to acknowledge Noelle, who didn't put herself on this slide, but she is my co-PI on the Knowledge Center. Okay, so let me look at questions here. Also, they have a data quality problem. That means, though, that we need to explain exactly how low quality data is found in. So what was this? I think I missed. What was this in with respect to when you what? Oh, LMMs. Yeah. So LMMs have like they do have a data quality, and they need to explain how. Yes. So that's, I think, one of the benefits that you would get from like a RAG-based approach, right? That you're feeding in LMMs like higher quality data that it can then use. And my experience has been when I ask an LMM to summarize data or text, it is much more careful and accurate than when it just gives me a question out of the blue, right? So I think that is a reason that that's a really good way to do it. The one thing I would say, to be a little bit, I wouldn't say contrarian, but I often hear this complaint like that knowledge graphs are necessary because LMMs don't have provenance and show their work and don't have high quality data. But as people astutely asked and questioned, in practice, knowledge graphs have a data quality problem too, right? So a lot of like the gene disease assertions that are going into Translator are, and that's why I put like this thing up here, are like from these OMIM like studies that are like 30 years old and no one trusts, right? And the people, and like, it's very, very hard in practice to get that quality up. So even if you, while in principle, you can get a high quality graph, in practice, we aren't there yet. And that has to be weighed aside that like LMMs have gotten dramatically better in the last year. And we just have to be clear-eyed that when billions of dollars are being pumped into that, they're going to be addressing that. And I think it's just important for us as researchers in the academic sector to be clear-eyed about where we can contribute. And hopefully I've made that case that if you invest in appropriate statistical ways to get your data in, you can solve the data quality problem with knowledge graphs. And now that percolates on and can improve LMMs as well, because now they're getting access to the high quality data. And who knows, maybe at that point the LMM's not needed, maybe it's together. Those are kind of orthogonal things, but that's kind of, so I agree with the data quality problem. But I do want to have people think critically that just because a theoretical knowledge graph is high quality, the practical ones I've worked with, as I kind of hope to illustrate, often are lower quality than we'd like them to be, even after this much investment. All right, I have 2 minutes left.
Yeah, are there any more questions? You can unmute yourself right now to join the discussion. Yeah. I guess most of them asked the question during your presentation. Yeah.
If they did, then I nailed the timing perfectly, which I'm very proud of. But oh, another question in the chat. Thank you. Okay.
Yeah.
All right, good. Well, I hope, like I said, I'm not a knowledge graph expert, so take everything I have with a grain of salt. You should view this as someone who's trying to use them. I'm interested in how to solve these biological questions with whatever technologies are available. And so, like, to me, it's not about like LMMs versus knowledge graphs. It's really like, how do you put them together to address these challenges? And I just want to, again, iterate. I think there's a huge opportunity and underinvestment in getting knowledge sources onboarded to these knowledge graphs. And I think that is a huge area where it's not going to get cannibalized by industry. And if we can figure out how to do that, that'll be a really big service to the field.